Okay, we've done the first section of the review, which makes up about half of your tests. What I'm going to do, I'm going to fold that back over there like that, and we're going to focus on this section here. Okay, so what have we got here? This looks a lot, I just pull my notebook up, a lot like this, right? If you recall, we did the karyotypes when we did a human karyotype where we matched everything up, and remember, they form homologous pairs, one from mum, one from dad, and so on this chromosome you've got DNA that carries certain characteristics. On this chromosome you've got DNA which carries information about the same characteristics, which may or may not be the same dependent upon whether it's homozygous for a certain allele or heterozygous. Okay, key thing here is they all come in pairs. So when you look at this one, where is the mutation? If you notice, you've got 2, 2, 2. Oop, what have I got there? I've got 3. Okay, so the mutation occurs at chromosome pair number 4. And this may be an organism that only has 8 chromosome pairs. Okay, remember, humans have 23 pairs. Other organisms have different numbers. What is it called if you've got 3? It is called, if you recall, trisomy. Okay, why do we get trisomy? Why do we end up with, for example, trisomy 21 is Down syndrome? Why do we get that? We have to go back to this bit, non-disjunction. Remember, normally they separate and one goes into each cell to make the 4 gametes. But here, instead of them separating, they both went this way, which meant I ended up with 2 in one cell and nothing in the other cell. And that was called non-disjunction, when both the chromosomes move the same way and they don't separate. So at fertilization, this is what should happen: single, single, gamete, gamete, fertilized egg cell. Here we have a single, normal. This is the trisomy, where we end up with 3, and then monosomy is where we end up with 1. So this is called trisomy. What is it caused by? It is caused by non-disjunction. Okay? All right. Here, this is a quick thing, and I made a little mistake here, so you need to change that number to from a 2 to a 3. Now, I put this in here because you're going to see this word. So anytime you have chromosomes 1 through 22, they are called autosomes. Okay? When we talked about sex-linked inheritance, autosomes are the ones which are not related to whether you're male or female. And sometimes you'll see that as autosomal. It just means that whatever the alleles are, whatever the genes are, they're carried on non-sex chromosomes. Chromosome pair number 23, those are your sex chromosomes. And remember, I'm just going to write sex chromo. You remember when we talked about sex-linked inheritance, if it's on chromosome pair 23, it's going to be inherited differently if you're a male compared to if you're a female. Okay? So I just wanted you to have those, because I don't think that autosome or autosomal was in our words page. Okay, these 3 terms here kind of relate to this, the monohybrid and the dihybrid crosses, because it is really important that if something is homozygous dominant, homo means same, so it would be the same allele. So big A, it doesn't matter what they are. You're going to have 2 capital letters. That's how you know it's a homozygous dominant genotype. Homozygous recessive would be, for example, little b, where you've got the same, again, homo means the same alleles, but they're both recessive this time. Heterozygous, on the other hand, means whatever you've got whatever the genotype is, whatever the allele is, you're going to have one dominant allele and one recessive allele. Hetero means different, which means you've got different alleles for that characteristic. Okay? All right, this next bit, this is one of my pick-me's. Comes up over and over again. And this doesn't relate so much to this unit, but if we go all the way back to Unit 4, we talked about cell differentiation. So these are things called pluripotent stem cells. They are cells which are undifferentiated. They haven't really decided what they're going to be, but they have the capability of becoming any type of cell that they need to be. And so if you have pluripotent, they could become muscle cells, blood cells, nerve cells, epithelial cells, skin cells, whatever type of cells you need them to be, because they have a complete set of DNA. So they've got the information to be all types of cells. So why, if they have all the information, do they become muscle cells? Well, they become muscle cells because this section of DNA here is switched on while everything else is switched off. But if you switch off the muscle cell and switch on the intestinal cells, these cells can grow into intestinal cells. This is really important when you've got things like skin cells. When they're trying to make skin cells, they can control which DNA. Like if you're a burns patient or something like that, they can take cells from one part of your body and use them to produce skin cells because they carry all the information. But what controls which type of cell they become, right, depends on which section of DNA is on or which section of DNA is being expressed. So you can take a stem cell and turn it into everything based on the regulation of gene expression. That is a very important phrase for you to remember. Regulation of gene expression. That's basically you're regulating which bit of the DNA, which genes are turned on, and which genes are turned off. Okay, so the next one here, we've got horse, 2n equals 64, horse sperm, n equals what? I wanted to put this in here just to make it clear to you that when we talk about haploid and diploid in humans, 2n in humans would be 46. 2n in humans would be 23. It doesn't matter. The rules are the same. It's just every organism has a different number of chromosomes. So in horses, their body cells, 2n equals 64, right? They have 64 chromosomes in all their body cells. But if you're a sperm cell in a horse, what is half of 64? It is 32. So horse sperm and horse eggs would have 32 chromosomes in each cell. Very simple, very obvious. Just didn't want anybody tripping up on that. Okay, so this one here is talking about your genetic code. And first off, complementary base pairing. If there is an A on one side of the DNA, we know complementary base pairing, there's going to be a T on the other side. If there's a G, it's going to be a C. If there's a T, it's going to be an A. If there's a C, it's there. We go, just like that. Okay. What is your genetic code? For some reason, this is something everybody gets hung up on. We know it's the order of ATGCs. Those ATGCs are also called nitrogen bases. Okay? Those ATGCs are part of the building block of DNA. And the building block of DNA are called those little things where you have a phosphate, a deoxyribose sugar, and the nitrogen base. That is a nucleotide. So when you say what is your genetic code, it is actually the sequence of nucleotides. Okay, it is the sequence of nucleotides. It's the order of the ATGCs, because remember, 3 nitrogen bases in DNA is a gene, and that gets coded for 3 nitrogen bases in messenger RNA, which is the codon, which then goes on to produce the amino acids that make the proteins, and proteins are our characteristics. Okay, all right, let's look at this one here. Okay, this is just a different way of looking at something that you are already familiar with. Okay, so up until now, if I put these side by side, this would not be an issue because you've done it that way. But it says here, what are the possibilities of independent assortment? The rule is the same as what we did up here. Okay, you have to have one of each letter. So I have to have, if that's a big A, I have to have one from up here and one from down here. So big A could go with a big E. That's one option. But a big A could also go with a little e, couldn't it? But I could have a little a and a big E. Or a little a and a little e. Okay? Little a, little. That is independent assortment. And if you can do that, you can do any dihybrid cross that there is. Okay, so that is the second section of the review. And we'll pick it up with